Before I come to the topic of my talk, I would like to say a few words about myself. We already mentioned I'm from DLR, the German Aerospace Center. The name is a bit misleading because DLR is not only working on aeronautics and space, but also on energy, transport, security, and digitalization. So it's a very broad topic and a very diverse group of institutes at DLR. In addition to these research activities, DLR has also one branch which is the space agency, like CNES in France, but it's part of DLR, and it's also a project management agency for different ministries. There are about 54 research institutes and facilities at 30 locations in Germany. DLR has more than 10,000 employees, and DLR is member of the Helmholtz Association of German Research Centers. You might know some others like the Alfred Wegener Institute for Polar Research or other centers working on cancer in humans. So I retired last year from the Institute of Aerospace Medicine. This is the only life science institute at DLR. And there have been two groups of microbiology, which are now combined with my colleagues. And the general topic, what we are doing is to look at the resilience, versatility, and adaptability of microorganisms to changing and extreme conditions. This is, of course, a very broad topic, and this is done also by many other research groups at universities and other institutes. However, the difference at DLR is that we are doing this under an astrobiological perspective. So what we are doing is really hardcore microbiology, but our way to formulate questions and how to approach investigations is different than many other groups who are doing a kind of astrobiology, but who are not aware to be doing this. So in our group, we are interested in three main topics: the interaction of microorganisms and environmental factors as they occur in space, for example, on our neighbor planet Mars, but also on the icy moons in the outer solar system. This is important for robotic space missions. But in addition to this, we are also interested in the interactions of microorganisms and humans, because for long-term missions of astronauts or for astronauts living on habitats on Moon and Mars, microbes might pose a risk due to pathogens which might accumulate. And we are looking to this because it's a very important topic for manned missions. Connected to this are the interactions of microorganisms in confined habitats. On Earth, we have almost everywhere open systems, but spacecraft and space habitats will be very confined, closed, hermetically closed systems. And there, the dynamics of microbes might be different than in open systems where we have an exchange with new microbes coming in, others dying out. And this is for manned habitats, spacecraft, but also exploration greenhouses, which are necessary for long-term missions to produce food for the astronauts. So our focus is microbiology under an astrobiological perspective, covering different topics. Overarching to everything is planetary protection. This is an activity which is done since the beginning of lander missions to Mars internationally, because when we are flying to other planets or moons, we have to make sure that we do not contaminate other planets and moons with our own microbes, identify them later and say, oh, we have found life on Mars. This is implemented since the Viking missions, but a lot of effort needs to be done and a lot of research is necessary to improve the existing planetary protection measures. But today I'm not talking about planetary protection itself. So we are here on an astrobiology summer school, and I think you have talked about this in general before, but I would like to repeat it. What is astrobiology? There have been a couple of definitions over the years. I think the best one is one from the US national academies of science, engineering, and medicine, shown here. Astrobiology is the integrative study of the interactions within and between the physical, chemical, biological, geological, planetary, and astrophysical systems as they relate to understanding how an environment transforms from non-living to living and how life and its host environment co-evolve. It's a long sentence, especially in the English language, but I think it's a very good description what astrobiology is. In the past, the term exobiology was used, but it has a narrower scope, meaning mainly looking for extraterrestrial life. But astrobiology in general, it's much more than this. Astrobiological questions are not new. They are very ancient, and some examples are shown here. How, when, and where did life on Earth emerge? What were the first forms of life? What were the environmental conditions at that time on Earth? What are or have been the environmental conditions on other planets and moons, and how did they change over geological timescales? Are there other forms of life in the universe? I think this is the question. How might it look like? Is it similar or very different to life on Earth? And very important for space missions, how can we detect it? If it's very different from our life, it might be hard to find it because you have only restricted number of instruments and type of instruments to look at some features. So these questions in general are not new. Already Giordano Bruno stated in the 16th century that he thinks in space there are countless constellations, suns, and planets. There are also numberless Earths circling around their suns, no worse and no less than this globe of ours. So the ideas of life beyond Earth are not new, but we are now in the lucky situation that we can address these questions by empirical scientific research. And especially in the last 20 years, or 30 years, there was really impressive in technology development, which helps to try to answer questions about extraterrestrial life. There was an amazing progress in biotechnology, but also in space technology. And we know much more about life in general on Earth concerning extremophiles. We are working now on a more overarching approach in systems biology, and we know much more about ecology. So we are now in the situation where we can address these important and very exciting astrobiological questions by doing scientific empirical research. And to do this, it's necessary that a lot of disciplines work together. Some of them are shown here. This is from the webpage of the European Astrobiology Institute, but there are many subdisciplines more, and you need all of them, from astronomy, chemistry, biology, geology, planetary science to space technology to answer the questions I've mentioned before. This is my personal opinion, one risk is when you start to do astrobiology, you have to come from at least one discipline. You have to be really an expert in one discipline, but you have to learn more from other disciplines. And sometimes some people have the impression, oh, I do astrobiology and I know everything from all these disciplines. You should not forget you are not an expert in all of them. And you have to learn how to work with the others together. And that's a long process. It never ends, and it's the interesting part. So what do we know about life as we have it on Earth? We have requirements for life. One is liquid water. Water can or is used as a solvent, but also as a reaction partner in biochemical reactions in cells. But we also need to have chemical elements to build up the components of the cells and of the whole organisms. The most important ones are, of course, carbon, hydrogen, nitrogen, oxygen, phosphor, and sulfur, but some others in minor concentrations. And we need a light source. On Earth, the biosphere we are looking at as humans are based mainly on the energy source from our star, from the sun, by photosynthesis. Either organisms are able to do photosynthesis or organisms are using those which are able to do photosynthesis. In other words, we are eating plants if we cannot do photosynthesis by our own. Independent from this, you can also organisms can also use chemical energy, and this is done by a lot of microbes, and many of them are the extremophiles I'm coming to. So what we know from life on Earth helps us to define habitability, and this is a measure of a planet's or moon's potential to develop and maintain environments hospitable to life. It's, however, the understanding of what habitability is an extrapolation of the conditions on Earth, because Earth so far is the only planet which harbors life. It's so far a specific point of view to define habitability based on life on Earth. And we shouldn't forget, on the early Earth, when life emerged at least 3.4 billion years ago, the environmental conditions on our planet were quite different from those which we see today. Nevertheless, the concept of habitability is necessary to plan for missions for life detection. And our knowledge about the physical and chemical limits of life on Earth is the basis for habitability and supports the search for habitable environments in space. However, habitable does not mean inhabited. Habitability of an environment can change over geological timescales. Habitability can be restricted to certain areas. It must not be planet-wide. Habitable niches can be quite small and therefore might be difficult to detect by space instruments. And habitable regions on a planet or moon might not be accessible by in situ instruments. So searching for life in our solar system is not that easy. There was a recent publication from the end of last year by our colleague Charles Coquelle from Edinburgh. And if you are more interested in the concept of habitability and more theoretical and philosophical considerations, I recommend to read these articles where he talks about uninhabitable environments, habitable environments which are not inhabited, and habitable environments which are inhabited. And each environment can transform into another by different processes, and this is a quite interesting, as I said, more philosophical paper about these conditions in hypothetical environment types in the universe. And from the same paper, there is one example of potential uninhabited habitats at a macroscopic scale on Mars. On the surface, there is no stable liquid water due to the actual environmental conditions. However, there might be liquid water, for example, by a meteoritic impact or induced by a space mission with radioactive material, which might help to melt the frozen water in the cryosphere. But if there is no exchange between any subsurface which might be inhabited and these transiently liquid water bodies, you will not find anything because they remain sterile. And these things look a bit sophisticated here, but it's really important to think about it in detail when you are trying to plan a space mission, when you are trying to plan drilling. You have to be really aware of the environmental conditions and try to have the best choices where to land and what to do there. So in our solar system, we have a couple of habitable environments which are interested for investigations in prebiotic chemistry and the search for extraterrestrial life. As I mentioned, the most interesting and mostly to be investigated planet is our own Earth, But Mars is also very interesting, the present day Mars, but also the past Mars, and some moons in the outer solar system around Jupiter and Saturn, which are covered by a frozen water ice layer. And beneath this water ice layer, there are liquid oceans with salty water. And here, especially interesting are Enceladus and Europa, but also Titan, another moon of Saturn, is very interesting because of the chemical reactions in Titan's atmosphere, which contain very interesting compounds. And I think you might have heard about this during the summer school. The search for signs of extraterrestrial life is one of the major drivers for solar system exploration. And this is reflected by the several missions which are ongoing or which are in preparation. Here are just two missions I would like to mention for Mars. There's the Mars Sample Return Campaign from NASA and ESA. Even if there is a new restructuring ongoing now, I'm quite sure it will happen. The first step is Mars 2020, which is successfully already on Mars collecting samples. And these samples, about 30, will be brought to Earth with later missions with the European Earth Return Orbiter, so that these Martian samples can be analyzed on ground with the best instruments and best technologies and the best knowledge at that time. Another, for us, interesting mission is ExoMars from ESA. There were many delays due to very many different reasons. The reason for the last delay was the Russian war in the Ukraine, so there had to be a complete replanning of ExoMars. But now the launch is scheduled for 2028, and we are quite excited because there will be a drill which should be able to drill up to 2 meters and analyze samples in depth from Mars. But these are only two examples. Many other agencies are planning and preparing space missions. For example, the Chinese colleagues are also preparing, amongst others, Mars sample return, and they might be even faster than NASA and ESA. I mentioned the icy moons. There is an ongoing space mission from ESA called JUICE. It was launched in 2023. And it has an orbiter which will analyze especially Ganymede, Callisto, and Europa. A bit later, the NASA Europa Clipper mission was launched last year, which focuses on Europa. And also the NASA mission Dragonfly is in preparation, which will go to Titan. And part of this mission will be a kind of helicopter flying around and doing analysis on the Titan's surface at different places. But as I said, these are only a few examples. There are many more international missions looking for signs of life which are in preparation or ongoing. So life on Earth, the only inhabited planet we know of, can be very diverse. There are many very small living organisms, these bacteria and archaea, which are in the micrometer scale. There are a bit more complex organisms which have a cell nucleus, which are a bit larger, but we also have lichens, plants, animals of a whole variety of species, including humans. Life on Earth is very diverse, but it's very hard to define what life is. And there are different attempts to define what life is. And here are some of them. One is living organisms undergo metabolism, maintain homeostasis, possess a capacity to grow, respond to stimuli, reproduce, and through natural selection, adapt to their environment in successive generations. Another attempt to define it is more from a physical-chemical point of view. Living organisms are self-organizing, dissipative, non-equilibrium systems with the ability to evolve. In the strict sense, most of the attempts to define life, it's not really a definition, but a list of features and characteristics. And most of them can also be found in non-living entities. Therefore, there is no real universal definition of life accepted so far. Life on Earth is present almost everywhere in so-called moderate and extreme environments. The question is, what do we mean when we say extreme? This word extreme is from an anthropogenic point of view. For us, it's normal to have 21% oxygen in the atmosphere. A temperature of plus 20 degrees is great, and pressure of 1 atmosphere is great. But for many living organisms, it's not optimal. So extreme is an extrapolation from us, and it's really anthropogenic. For organisms living in what we call extreme environments, for them it's normal and they need these environmental conditions. The definition of extreme environments is also not really clear. One attempt to define it is shown here. A given environment where one or more parameters show values permanently close to upper and lower limits known for life may be considered as an extreme environment. That's also very vague. Extremes can be concerning pH, salinity, the presence of high concentrations of oxidizing compounds, temperature, pressure, radiation, composition of the atmosphere containing oxygen or being anoxic, water availability, availability of organic carbons, and many others. Prokaryotic and small eukaryotic organisms, here archaea, bacteria, and simple fungi, are the dominating forms of life in extreme environments, and therefore these organisms are called extremophiles, living or loving the extremes. So on Earth, we have a really amazing number of microbes, even if we are not really aware of this. This cartoon is from a publication from Fleming and Würz from 2019, and they estimated the total number of microbial cells on Earth, and they came up with an estimation of 1 × 10⁻30. This is a really number you cannot really imagine, and the majority of the habitats of these microbes are not really visible to us. Many microbes are living in the soil. Many are living in the deep continental subsurface, in the deep oceanic subsurface, in the upper oceanic sediments, and in the ocean itself. And what we see as biosphere as a human are only minor habitats for these microbes. And all these things about extremophiles in extreme environments were not known 40, 50 years ago. These are found in the last years by new technologies and new attempts to look for the limits of life on Earth. Here is a kind of cartoon showing a cross-section of Earth's crust and the diversity of extreme environments and where they are. There are environments on Earth with low temperatures like sea ice, permafrost, and polar regions. There are environments which are anoxic, for example, in the deep subsurface, but also in deep sea anoxic lakes and brines, for example, in the Red Sea, but also in the Mediterranean Sea. There are environments which are very hot, for example, in the deep sea hydrothermal vents, where the temperatures can be very, very high. But also on land, you have hot springs, fumaroles, but also mud volcanoes. Other environments have a very high salinity, where you have very high salt concentrations with different salts. Some of them might be toxic for a couple of organisms. You have environments which are very acidic or alkaline. You also have environments which are very dry, like cold and hot deserts. And you have also artificially induced extreme environments due to human activities. For example, acid mine drainage, where you have a very low pH and very often high concentrations of metal ions, which are toxic for many organisms. Another example for human-made extreme environments are nuclear contaminated sites, where also a lot of microorganisms can be found. All of these environments are inhabited by extremophilic microorganisms. But in most cases, not only one parameter is extreme, but more than one. And therefore, a couple of model microbes are named polyextremophiles, meaning loving different extreme environmental parameters at the same time. So those, and they have specific names, those who are living in the cold are called psychrophiles, cold loving. Those who are living in hot environments are called thermophiles or hypothermophiles, also heat loving. And you have these others, xerophiles, adapted to desiccation conditions, piezophiles, adapted to high pressure conditions, halophiles, requiring high salt concentrations, or anaerobes, which grow in the absence of oxygen, and for most of them, oxygen is toxic. These are a couple of examples, and it's really amazing which metabolic features are present in different organisms in these extreme environments. And our knowledge about the physical and chemical limits of life on Earth can be used to support the search for habitable environments in our solar system. Here's just one example from a paper from Merino from 2019, showing here only 4 parameters: temperature, pressure, pH, and salinity observed for life on Earth, compared to the same parameters observed in other interesting places in our solar system. And you can see there is a big overlap in these parameters, and these are only 4 of them. And it's also an interesting publication I really strongly recommend to read. Coming to astrobiological research, it can be very different depending on what scientific questions you want to ask. You can do field research in space analog sites. On the picture in the upper left, you can see colleagues from a project and myself doing sampling in an Icelandic lake, which is quite acidic and very cold. But most of our research is done in the lab, in normal microbiological labs. For resistance tests, we are using space simulation facilities where we try to mimic the environmental conditions in space or on the surface of Mars in our facilities as good as possible. Of course, it's not perfect, but for prepare space experiments, it's absolutely necessary to do these kind of tests in space simulation facilities. We are also supporting the development and test of life detection instruments, because you need to know the capabilities of your instruments and you have to prove it with your own microbes you know about. And we are doing experiments in space, here so far only in low Earth orbit. I would like to give you one example of our recent work in the lab. It's done by my PhD student. That's part of his PhD thesis, Tommaso Zaccaria, and it's what I'm presenting here is accepted for publication. In this project, we want to investigate the survival limits of cyclotolerant microorganisms, which are relevant for planetary protection of the icy moons. We have to avoid that we contaminate the icy moons before we have investigated them, and we have to make sure that we do not bring organisms with us with the space missions which lands on these icy moons, which might be able to survive and multiply there. And in this case, we started to work with microbes isolated from extreme environments on Earth and try to find out if they can survive a travel through space and if they might pose a risk for planetary protection. I don't want you to read everything, just to show you. We started with the optimistic idea to have one model organisms, at least one from the group from the 3 domains of life. Here from bacteria, archaea, and eukaryotic cells. The blue ones are bacteria, the green ones are archaea, and the magenta ones are yeasts, eukaryotic organisms. Most of them grow at very low temperatures. Just one example, the first one, Planococcus, grows up down to -10 degrees. and one of the yeasts grows down to minus 15 degrees centigrade. However, they can grow at higher temperatures and they grow faster at higher temperatures. And we obtained these microbes from culture collections, but they were originally isolated from the Arctic permafrost by our colleagues from Canada. One was isolated from a very saline lake in Bolivia, others from Antarctic soil, from sea ice, from permafrost in Tibet, from a very interesting soil-containing lake in China, but also from a deep sea brine pool in the Red Sea, from Antarctica, and the last one was isolated by us from a clean room where spacecraft have been assembled from Toulouse. We wanted to investigate these organisms, but we had certain criteria to select them. First of all, they should be able to grow on solid nutrient media. You might know this, Petri dishes. They should be able to form colonies. Not all of them are doing this, especially archaea don't like this. Then we wanted organisms which are able to grow without oxygen, at least facultatively. And we wanted organisms which show visible growth in 4, maximal 5 days. Otherwise, you can't do these kind of experiments in a 4-year project. It takes too long to get results. And this is one reason why there is not so many informations about psychrophytes. In the end, we ended up to investigate only 4 of them, and we didn't find a suitable archaeon which filled in our criteria. We assume that the composition of the icy moon's ocean is very nutrient poor, meaning oligotrophic conditions, and we have chosen a medium which contains only salts and 1 carbon source in a concentration of 0.2%. This is a very low concentration, and we tested different simple organics, which also have been identified in carbonaceous chondrites, and which might have been brought to Earth, to Mars and other planets by meteorites, for example. And we started to test alanine, glyceric acid, gluconic acid, and glutamic acid. And for controlled reactions, we used glucose, which is part of the normal metabolisms of all cells. I don't want to go into too much detail, just to show you what kind of experiments we are doing. We assume if a microbe is of concern for planetary protection, it has to be able to survive desiccation over long periods of time, because a space mission to the outer solar system takes at least 7 or 8 years before the target moon is reached. And therefore, we tested the desiccation resistant under different conditions, under oxy conditions in air, but also under anox conditions. In space, we do not have oxygen. At different temperatures, +20, -20, and -80 degrees. And as you can see, just looking at the colors, the survival is quite different depending on what organisms you are looking at. All these data have not been known before, and we were quite surprised that especially the yeasts, and especially the one from the Arctic, is really quite desiccation resistant. Here we tested up to 160 days. When the curve stops, the plot stops, it's either there was no survival anymore or we didn't have any more samples. In the middle, you can just have a look at the resistance to freezing without any cryoprotectants. And here also the yeast from the Arctic is very, very resistant. It's not influenced at all by freezing. We also looked at the freeze-thaw cycles. We cycled between -80 and +25, and just now we are at cycle number 200. The work is ongoing. Here, to our surprise, the second yeast we looked at is very resistant. After a first decrease in survival, it's more or less the same since 130 cycles. And this yeast is interesting because it was isolated from the clean room in Toulouse. So it was really a living cell found in a clean room where spacecraft are assembled. And this might be really a concern if we didn't nevertheless, we didn't expect this because this yeast is human associated. It's not psychrophilic, and it can also cause some health issues with humans. In addition to this, we looked at the radiation resistant concerning UV radiation, but also ionizing radiation. And again, the two yeast strains turned out to be quite resistant. And from this, I can conclude that yeasts are, or fungi in general, are not really considered so far for planetary protection, and there should be more monitoring and investigations to find out if yeasts might be a bigger problem than bacteria and even archaea. So now it's an ongoing project. We are looking at the combined exposure to these different parameters, desiccation, ionizing radiation, and UV radiation, as it occurs on Mars. And we have exposed these yeast cells. And after that, we allowed them to repair their damages inside the cells for different periods of time. And we are looking at the survival and doing transcriptomics to find out what the cells are really doing, what the processes are, what genes are turned on, what genes are turned off, and how they are able to survive these combined exposure to these extreme conditions. And again, this yeast from the Arctic is able to grow in minimal media with glutamic acid as sole carbon source, and it can tolerate the combined exposure to icy moon relevant conditions. As I said, the transcriptomics results are not yet there, but they will be there soon. And I think it's really exciting that yeasts are so resistant. Coming to space, we are also we astrobiologists are all using space as a tool for doing experiments. And this kind of research was initiated long time ago by Gerda Horneck and Günther Reitz. Gerda Horneck both from DLR, from our institute. Gerda Horneck doing microbiology and Günther Reitz doing the dosimetry of ionizing radiation, which is very important to know. And the very first astrobiological experiment was done already during the Apollo 16 mission in 1972. And you see on the left side a picture of the exposure facility. It was more or less quite small and was mounted on the Apollo capsule. And this was really the very first European, as a general, but also European astrobiological experiment in space. Later on, it was possible to do exposure experiments in space for up to 2 weeks on space shuttles. The samples were accommodated in the cargo bay of the space shuttles, and in orbit, the cargo bay could be opened, as you can see it here on the picture on the left side. And your samples were exposed to the space environment. And if you wanted to investigate UV effects, the shuttle has to go into a certain attitude so that the cargo bay was oriented to the sun, also in zenith orientation. In parallel, it was possible to do experiment on the Russian space station Mir, which was available at that time. For example, the French colleagues had a mission exposure facility called Perseus, but it was also possible to do experiments inside the Mir station. Another still very often cited examples is the use of a free-flying satellite called LDEF, Long Duration Exposure Facility. It was planned to be in space for 6 months in 1984. And then there was the first shuttle accident, and only 6 years later, the satellite could be retrieved. And microbiological samples were placed on the satellite, as you can see where the black arrow is. In the 90s and at the beginning of the 21st century, we had the opportunity to do a couple of experiments on Russian satellites using the European facility BioPen. And this is what LV was talking about before. This is where our collaboration started already. This BioPen facility was a pen-like structure. You can see it in the middle picture in the upper part. It was opened in space. The samples in both sides were exposed to the space environment. Then it was closed. and brought back to Earth with a landing in Kazakhstan. This was really a good way to do experiments because we could have it every 2 years, and it was straightforward and it worked quite well. Since many years now, more than 30 years, we have the International Space Station. And there it's possible to do long-term exposure experiments. There were 3 European EXPOSE missions called EXPOSE-E, R, and R2. Their samples were exposed to space conditions for up to 2 years. As you can see here on the picture, EXPOSE-E was mounted on the European Columbus module outside on a platform. EXPOSE-R was mounted on the Russian module, and EXPOSE-R2 at the same place, but a couple of years later. Since 2015, also JAXA, the Japanese space agency, and the scientists in Japan are doing long-term exposure experiments in their hardware on their module in the Tanpopo missions. And we collaborate to a certain degree with our Japanese colleagues. These are ongoing, and now they are at the Tanpopo, I think 6 or 7 mission right now. To show you how this can look like, these exposure facilities, this is EXPOSE-R. It's a monoblock. It has 3 trays with 4 compartments each. The compartments are connected by a venting line because many samples should be exposed to space vacuum. And the valves were opened by telecommand in space and closed again before the hardware was brought back to Earth. In addition to these exposure places, we had dosimeters for UV radiation, active dosimeters delivering data about the UV fluence the samples obtained. We have had different sample carrier types. I show you in the next slide. We had temperature sensors at different places because there was a gradient over this facility, which is about 50 centimeter in length. And we had one instrument to measure the ionizing radiation. Just to show you the kind of experiments which were flown at the same time during this EXPOSE mission, there were 3 biological experiments investigating a variety of organisms, bacteria, archaea, lichens, mosses, fungi, yeasts, but especially our Russian colleagues exposed also plant seeds, mosquito larvae, triops, and many others. In addition, there was the group of chemical experiments from Hervé Cotin. They looked at different chemicals, and if you want to know more about this, you should ask him. The active UV dosimetry was done by an instrument delivering data during the mission and sending it down to Earth. And passive dosimetry was done by TLDs from our institute. And in this mission, we have had a total number of samples of 696 from different groups because all of these experiments had a lot of colleagues contributing to one experiment. So it was a lot of logistics to organize these 696 samples. We have had one tray, which was called space. So in this tray, the samples were exposed to space vacuum after opening the valves. The samples were covered by magnesium fluoride windows, which allow the transmission of solar UV radiation down to 110 nanometer. And this layout was according to what the scientists wanted to have. The upper layer was exposed to the sun, and the 1 or 2 lower layers are used as in-flight dark controls. One tray was called Mars. There we have had closed sample containers containing Martian atmosphere and pressure. And these samples were covered by quartz windows, which allow the transmission of solar UV radiation down to 200 nanometer as we have it on Mars. And the third tray was also a space tray with vacuum and magnesium fluoride windows from Hervé's experiments. Here you can see a picture how it looked like in reality. You see part of an astronaut because astronauts have to go outside of the space station in an EVA, extravehicular activity, and mount this exposure facility outside, and after 1.5 years, they have to bring it back inside the space station before it can be downloaded to Earth for analysis. It's very important that you have a very good preparation before the space flight. You have to do a lot of tests to find out the optimal conditions for your samples, because you have many restrictions, and it's utmost important to specify the experimental parameters. This is done, of course, by your own, but also in a combined way during the so-called experiment verification tests to show that in principle, your experiment will work in space. And in addition, you also need an experiment sequence test to simulate the sample assembly, the parameters to do this, the disassembly, and you have to have an eye on the time schedule. If you have 696 samples, you have to make sure that you know where they are, under which conditions they have to be stored before you can mount them into the hardware. This has to be done in a very coordinated and controlled way, and you have to document every step. And this is really important, otherwise you are lost when you get the samples back after 1.5 or 2 years and you do not know really what you have. It's also very important to do always a parallel mission ground experiment with identical samples in identical hardware exposed to the parameters which are downlinked from the satellite or space station by telemetry, to make sure that you avoid any artifacts which might occur during some unwanted issues during transport, for example, temperature or so, or humidity. And you have to really make sure that you know what you have in your lab under optimal conditions, and that you are able to interpret your results from your space experiment. And this is really important, and I think many people are not aware about this. This also means that you have twice the number of samples, one for the flight and one for the mission ground reference experiment, plus at least one set of samples as what we call lab control, just stored in your lab. So the results of these exposure experiments were published in, for example, 3 special issues in Astrobiology and the International Journal of Astrobiology, but there are many other publications in different journals reporting about results from exposed missions, and there are still, after some years, ongoing activities and publications coming out. Just recently, a new one will be published soon from the exposed missions. I think for you it might be interesting to find out how to do a space experiment. So you can have great ideas, but you have to find a way how to do this. And in our case, we are doing most experiments with ESA, the European Space Agency. And in the future, we will use the new Exobio facility, which is being developed and which will be placed on a Bartolomeo platform on the ISS. But there are also national experiments, and I would like to mention IR COSTA from RW. This was or is part of the Euro Material Aging project by CNES and ESA, and this was launched successfully end of last year. And it seems to be that national projects are faster than ESA projects. Might be simpler to organize. Concerning ESA, if you want to do a flight experiment, you have to look for announcements of flight opportunities and calls. And in these calls, there is a detailed descriptions of the conditions for space experiments. And if you think for your scientific questions, the conditions are valuable, you have to write a proposal to ESA specified to a specific call. And ESA sends all proposals to experts, international experts, to evaluate these proposals with respect to scientific merit. So if it's a really good scientific idea, do you have the capability? Do you have proven to be able to do what you say you want to do? And each proposal gets a specific score. If your proposal is higher than a given threshold, there is a second evaluation by a technical committee. And they have to find out if your proposal or if your experiment you proposed can be done, which if is it feasible to do it under the given conditions. You can have the best ideas and the best technical solutions, but if it's not possible to be done just now on the space station, you get a low score. And also here, if you have also here a high score, your experiment will be part of a pool of potential ESA space experiments. So this is only the first step, and this is not the guarantee that your experiment will fly, even if it's in the experimental pool of ESA experiments. ESA tries to accommodate as many space experiments as possible, but ESA also has to identify flight opportunities. And if they have done this, also the national ESA delegates have to vote for implementation because there are many more proposals than possibilities to do the experiment. So there is a high competition of already selected proposals. If ESA identifies a flight opportunity, it contracts industry for designing and building experiment-specific space hardware. And ESA provides hardware and supports experiment performance in space. The science team, that is you, has to define the requirements in constant exchange with industry for your specific experiment. You have to test hardware models. You have to prepare and deliver the flight samples. You have to perform lab and ground control experiments and finally analyze all samples. However, the financing must be provided by the respective ESA country. It's not funded by ESA itself. ESA only provides hardware and supports the performance in space. And this can be challenging. So space experiments are exciting. However, they can take a very long time. Here are just some examples from the experiments which are in preparation now from my former group, also which are in preparation now, just to show you how long everything can take. One experiment I will talk about a bit more is called Lux in Space. The call was from 2014 and the launch was scheduled for April 3 this year. So more than 10 years later. Another experiment in preparation is called Ice Cold from my colleague Elke. It will fly in the mentioned Exobiologie facility. Launch might be in 26 TBC. Another one from 2020 looks at the bacterial conjugation in space in microgravity. This will be inside the International Space Station using live cell imaging with the Flumias microscope. And the fourth one is called MaxSam, also from the AO in 2014. It is foreseen also for the Exobiologie facility, but it will fly in the second batch of experiments after Ice Cold and the others. So it's a very long-lasting process from a successful proposal to the real mission. And you can be cancelled on all stages. And therefore, I would like to spend some words about Lux in Space, because for me, it's the most actual one. In space, we have two very unique environmental factors. That is cosmic radiation, which is very complex, with different kinds of radiation, different energies, and different biological effects. And we have a microgravity, that is a lack of gravity or reduced gravity compared to Earth. And both factors have a lot of consequences in cellular metabolism and in the physiology of all organisms, from bacteria to humans. And in our experiment Lux in Space, we want to investigate the effect of microgravity on the enzymatic repair of radiation-induced DNA damages in bacteria. We are using a bacterial biosensor, and these cells react with bioluminescence emission to DNA damage in a dose-dependent manner. So you have a cell culture of genetically modified bacteria. They were damaged, in this case, by radiation. And after a certain period of time, they start to emit light. And the higher the damage, the more light is emitted. And this is called bioluminescence. And for this, we want to use the BioLab facility in Columbus. This is on the left side, you can see the Columbus module. On the right side, a picture of the biolab facility. There are different compartments, and we are using a compartment which has also a 1G centrifuge. And we have samples in parallel in microgravity and on the 1G centrifuge. To show you the challenges, the launch was scheduled for April 3. This is very soon, and it should happen on SpaceX 32. To do this, a team from industry of 5 to 6 people had to be at the launch site at least 6 weeks before launch to start everything for the launch campaign. From the science team, from us, 2 people had to be at the launch sites 10 days before to start cultivating and sample preparation. So it's a lot of planning, documentation, customs, and everything. Last week, as I said, launch April 3. Last week, we were informed from ESA that this experiment has been moved off SpaceX 32. There might be an alternative, perhaps in June, with a Cygnus rocket, but this has some technical issues. Or the next opportunity might be SpaceX 33, currently scheduled for August. So again, booking, planning, we are not working on one experiment alone, but all of us, industry and all the scientists, are working on different experiments at the same time. And if you have to prepare, you have to book again, you have to shift everything, then holidays season comes in. It's really challenging to do this because you cannot change the conditions. You are dependent on this. So to summarize, I would like to say again, space experiments are exciting, but there are a lot of challenges. You need national funding over a long period of time. You have to have plans for knowledge management and transfer. If it takes more than 10 years, you cannot tell a PhD student just do something or a postdoc. You have to make sure that what knowledge, expertise was obtained remains in your group, and that you do not have to start from the sketch every time. Another challenge are all the acronyms. When I started to work at DLR, I attended the department meetings, and during the first meetings, I couldn't understand a word what they are talking about. In space experiments, you always have limited sample mass and size. You always have limited energy for active instruments, and you have limited data transfer if you are online monitoring something. And you have to argue and really make clear that you really need these data transfer, energy, and also sample mass and size. An issue is the number of sample replicates. I know that some colleagues are happy with only 1 sample each. To my opinion, it's absolutely necessary to have at least 3, better more replicates, because in biological experiments, there might be some variations by itself, and it's really important to be able to publish later on to have enough replicates. It's almost impossible to repeat a space experiment. On Earth, you do an experiment, and if it gives good results, you repeat it to make sure that it's really reproducible. But in space experiments, it's almost impossible to do this. And therefore, even more important is the number of sample replicates. What's also important is interdisciplinary communication and understanding. So it's amazing which kind of sample questions you get. Here are some real questions which were asked to me. How much science is lost in percent if your culture is incubated at 25 degrees instead of 30 degrees? They wanted a number as an answer. And as a scientist, I can tell you, it depends. As I've shown before, if organisms grow optimally at 30 degrees, they are able to grow at 25. However, it will take longer. And you cannot say 20% of science is lost at 25 degrees. It depends on when you have this 25 degrees, if you would be able to prolong your incubation time, which is very often not possible due to automatic sequences in space. Also, it's really a communication thing to talk to engineers and mission planners what to do if something goes wrong. Another question How much time is lost in percent if the maximal storage time is 3 months and 1 week instead of 3 months due to a launch delay? Hmm. Again, you are saying it depends. If it's good for 3 months, it might be okay for 3 months and 1 week, but samples will not become better with time. Or just a recent example, what if there is a power loss for 1, 2, or 4 hours? Again, it depends. If the power loss occurs, for example, in the Lux experiment, in the first incubation period, it doesn't matter. If it's in the second, if it's in the third, it depends if it's after or before irradiation, and if all is recorded and followed up, and if the incubation time can be prolonged. But this is an answer which is not wanted by the engineers because also the people doing mission operations had to decide. There's a power loss for 2 hours. You say your science is lost, then we skip the experiment. Of course, you hope to get some results of it and you do a lot of compromises, but nevertheless, it's a challenge to do this. All these things are accompanied by a lot of formal documentation and reviews, and you depend on the schedules of many other involved parties. There are different ESA departments involved in one mission. You have to talk to different parts of space industry, even from the same company. You have to talk to the operations people from the microgravity user support centers, who are also, of course, working on many different missions in parallel. You are dependent on reviews from ISS partners, for example, for safety concerning toxicity, but also biological safety. You are dependent on launch opportunities. As I've mentioned before, there was an issue with the Cygnus rocket, and now our experiment is cancelled for SpaceX 32. There are others. You are competing with other ISS experiments, other experiments in the same facility, like the BioLab facility. The experiment before from colleagues from Belgium was delayed, so we were delayed. And you also very often need other facilities, for example, for cooling and storage, and they have to be available. And what becomes more and more important is there is a limited lifetime of the ISS. And as you have seen, it takes a long time from a proposal to a space experiment. And there are not many opportunities now to write a proposal with the idea that it can be flown as long as the ISS is in orbit. So a summary, what do you need to carry out a space experiment? You need a very good scientific hypothesis for a space-related question of broad interest. It should be something which can only be done in space. Otherwise, it's much, from a scientific point of view, better and it's easier and faster to do it on ground if you can do it on ground. In addition to the scientific idea, you need a good idea for how to do this in space. You do not have to make suggestions to engineers how to build hardware, but you have to have a clear idea of the requirements and limitations and the compromises you might be willing to accept when you start to plan a space experiment. You need interdisciplinary communication skills, I mentioned before. You need a suitable working environment. A small corner in a microbiological lab is not enough if you do these tests with the different hardware model, and so you need space and an environment where you can do it over a long period of time. You need colleagues with a broad expertise, not only the same as yours. You need, as I mentioned, knowledge management and knowledge transfer. Tests are repeated because people forgot that others have done this before in the lab. You need patience, and you need perseverance. I mentioned the Lux experiment. The proposal was from 2014. However, the very first time this experiment was proposed was in 1996. And in between, there were several questions from ESA, and they asked us send in a new proposal, and all the time it was a new evaluation, a new very good score, a new plan to start this experiment, and now we hope to fly this year. And you need funding for an unforeseeable long period of time. But I don't want to discourage you. Space is really great, and space experiments are really excited, and it's big experience if you see the rocket launches and say, this is my experiment which is flying to space.